Good afternoon, everyone. We are ready to go. This is Dr. Tim Williams, the Chair of Medicine here at the University of Louisville, and we have the privilege this afternoon of hearing from Dr. Anna Yang. She'll be talking about unraveling biological aging pathways in COPD. And to give her a proper introduction, please welcome our division chief and our stellar MICU expert, Dr. Mohammed Saad. Thank you so much.
Thank you, Dr. Williams. Good afternoon, everyone. It's actually my pleasure today to welcome and introduce Dr. Yang. She is one of our, hopefully, upcoming faculty candidate. Did her college at Tulane, followed by medical school in Australia, and then residency and fellowship at University of Pittsburgh. Currently, she's clinical instructor at University of Pittsburgh on her research track. She's a clinician scientist, and we are, of course, hoping to kind of attract her to come here and work with our team and continue her work. Of course, she's interested in lung aging and COPD, and I'm sure we're going to hear more from her today about her work. But she's a promising scientist, and I think you're going to hear a lot of her over the next decades because she's very, very smart.
Thank you for the very kind words, Dr. Saad, Dr. Williams. And thank you for inviting me to be here. Apologies again on being late. I got a little bit mixed up in between all the buildings. Got my steps in, but here I am. So I'm going to share my work on unraveling biological aging pathways in COPD. I'm on an institutional T currently. Just a quick overview, because I know we're starting late. I'm going to go over a little bit about just what COPD is, COPD multimorbidity and aging. Some of the work that I've done investigating multimorbidity and mortality in COPD, as well as some more mechanistic work looking at microbial translocation and receptor afferent A2. I know I'm talking to a room of really smart folks, internists, PhDs, but for those of us who aren't pulmonologists, what is COPD? It's really a heterogeneous disease. You know, I think back in medical school, we remember the phenotypes of pink puffers, blue bloaters, but really I want to emphasize it's heterogeneous. It's characterized by irreversible airflow limitation and distal tissue destruction. There's no cure, and it's a progressive disease over time. Why should we care? It's a public health problem. Globally and in the US, it is one of the leading causes of death. Prevalence increases with age, virality, lower socioeconomic status. It's a disease of disparities. COPD is also higher in women and non-Hispanic whites, although this is likely because it's significantly underdiagnosed. And COPD patients frequently die from non-pulmonary diseases, especially those with more mild and moderate disease, as you can see here in our GOLD 1 and 2 groups. And I want to highlight that actually you can see that cardiovascular disease is a significant mortality contributor here. And in fact, when we think about our patients with COPD, we recognize that they have often multiple diseases. This is a slide from the BODE cohort, multi-center longitudinal study that looked at comorbidities and mortality risks associated with them. All the comorbidities with significant risk of death are here in this black circle here. Prevalence is by circle size. So you can see that more common diseases like coronary artery disease, heart failure, and less common diseases like esophageal cancer as well were associated with a high risk of death in this cohort. And then moving on, I want to discuss how comorbidities also develop at an earlier age in our patients with COPD. So this is data from a Spanish cohort. At the top panel, you have the COPD cohort. At the bottom, you have the non-COPD cohort. They're age matched. Each band represents the number of comorbidities per patient. Darker shades are higher counts. And the reapers stacked are 1,000 deaths. So you can see that the patients with COPD here in the top panel develop these comorbidities about 10 to 15 years earlier and also die from the mortality rate is higher in these participants. So this has really led to a shift in which we think about comorbidity coexisting with other diseases to what we call a syndrome of multimorbidity on the right here. This reflects really chronic conditions with shared biologies, acknowledging shared risk factors and shared biology. So there are many contributors, of course, to disease across the lifespan, as shown here. The lungs are constantly exposed to the environment. So obviously environmental exposures like fat and smoke, air pollution, et cetera, are very important. But today I'm going to specifically discuss lung aging and accelerated aging. So to begin with, what do we actually know about aging and lung aging? Aging is the single greatest risk factor for chronic non-communicable diseases. And it's not actually just the number on your driver's license or what we would call your chronologic age. Biologic age reflects the accumulated systemic and organ-specific processes that reflect your biology and functional capacity. Specifically in lungs, we see loss of elastic recoil, alveolar surface function, aberrant inflammatory repair responses, and then especially loss of alveolar type 2 to alveolar type 1 cell differentiation, which leads to this remodeling, bronchitis, airway resistance that we see in COPD specifically. So I think one of my overarching questions is, how do we start thinking about biologic versus chronologic age, especially in the era of precision medicine? How do we quantify this, and how do we apply it to our patients? So to do that, I want to talk about how people have measured biologic age. On the left is a seminal figure from the 2013 paper discussing the hallmarks of aging. These outline fundamental biologic processes that drive aging. And then we have, starting in 2015, some more published methods to try to quantify biologic age. So we're going from more of a descriptive to a quantification process. The first clocks really investigated DNA methylation, then later generations added clinical parameters, and then now emerging methodologies are using omic clocks, omic changes, microbial changes across the lifespan, and markers of cellular senescence. And so this is kind of the basis for a question of, how could we potentially identify a lung-specific aging endotype that associated with multimorbidity and mortality in COPD. To do that, we analyzed data from COPD Gene. It is a multi-center prospective observational study of participants across the entire country. There are 3 phases now, I believe. And we looked at SomaScan and body composition data. And we looked at tobacco-exposed people, and we split them by spirometric obstruction or not. Just want to briefly mention what the SomaScan platform is. It's a high throughput proteomics platform, synthetic data-based molecules that selectively binds proteins. It's great for large cohorts, like the UK Biobank is another cohort that uses SomaScan as well as spiromics. It can measure thousands of proteins across a wide range with very high specificity and sensitivity, primarily optimized for plasma and serum. Previous applications have included biomarker discovery and target identification for pharmacodynamic monitoring in drug development. So I'm going to get into a little bit of the computational work that we've done. So on the left is the computational work, and then on the right are kind of the results that I'm going to discuss here. So we decided to use the Lasso model to kind of reduce the dimensionality of the 1,300 plasma proteins in the SomaScan data. So we used an adaptive penalty. We were able to reduce the proteins down to about 87 proteins. The model fit was 77%, and our Pearson correlation coefficient with chronologic age, or their age at visit, was 0.88, which is actually on the higher end of compared to most of the biologic age measurements that I discussed in the background. And so we're going to call this our biologic age endotype, this 87 protein signature. Then we want to make sure that, which, sorry, you can see is in the top graph there, of course. Then we derived a new variable called accelerated age, which was a residual from regressing the predicted biologic age and chronologic age, because we're interested in the delta, really, between the biologic age and the chronologic age, because that's where we thought we would see where potentially the disease was. So here you can see on the graph here, I've shown you here that this accelerated age that we were able to derive is independent of chronologic age. The lowest curve here you can see is pretty flat. There's no really correlation there at all. And then on the right here is what we found from the residuals, the distribution of accelerated age. So here at 0 on this bell curve, these are folks who are aging as anticipated. Their predicted biologic age matches their chronologic age. To the right here, their predicted biologic age is higher than their chronologic age, and then vice versa, it's lower on the left. And so, you know, these are like the good agers. And I'm happy to take questions now or at the end, you know, raise your hand. So that's kind of the computational methodologic approach that we took. And then we used logistic and linear regression to look at our outcomes. Multimorbidity was lung function, FEV1. And then we also looked at imaging, quantifying pectoralis muscle density, aortic and coronary artery calcifications, emphysema, and then also some functional measures like the Bode index and distance walked on the 6-minute walk test, as well as mortality. Here are just some baseline characteristics. So of the 1,084 participants, the groups are split pretty evenly. You can see that the COPD cohort is a little bit older in terms of chronologic age at baseline than the non-COPD cohort. More former smokers, interestingly, and then also the expected spirometric changes at the bottom, where those with COPD have worse lung function and obstruction. So this is our 87 protein endotype. This is a heat map. Proteins are ranked by effect size. So the red at the top, the positive last cell coefficient is associated with higher predicted biologic age compared to the blue here. I'll just take a second so that you guys can look at that. But proteins span multiple functional categories and do cluster into coherent biologic pathways, which I'll get into in my next slide. Some of these proteins are novel, and some of them are pretty well established in the literature, which is nice for validity. GDF15, growth differentiation factor 15, is very well known and studied biomarker for aging. And then I'll highlight some others. Lumican is an ECM protein implicated and studied in severe onset early COPD. We have some other, inflammatory markers, CXCL, CLs, and then interestingly, a few proteins associated with osteoporosis and sarcopenia. So on pathway enrichment analysis, we really saw that cell signal and dysregulation was a core signal, as you can see on the left by count number, and also on the right ranked by significance. And then along with extracellular matrix remodeling, one of the proteins we evaluated previously. And then we also see this big immune response and inflammatory response as some of the dominant biological processes. So for the interest of time, I don't want to get too much into the weeds of the pathway enrichment analysis, and I'm going to shift a little bit to discussing biologic age in this cohort, and then also some of the outcomes that we looked at specifically. But after adjusting for chronologic age, interestingly, we found that the individuals with COPD demonstrated about half a year plus mean difference of accelerated aging compared to those without COPD. It's, I think, modest, but significant. And so here are those, this is a table of the multimorbidities that I was discussing earlier. So this table is split by COPD here and non-COPD with beta coefficients for size effect and standard error, and then you have the p-values for both here. So I'll go through the COPD cohort first. You can see that all of the multimorbidities we looked at were statistically significant, with the exception of the MMRC distance score. So I will pull out, I guess, so for our 6-minute walk test distance here in feet, what this means is that for 1 unit of accelerated age, there was a 15-foot decrease on the 6-minute walk test. And if you remember the histogram, most of the people most are clustered towards the center, but there is that distribution. So some of them, if you have 3 units of accelerated aging, that would be 45 feet. And then we see a decrease in lung function. We see an increase in emphysema, corner aortic calcifications, and decreased muscle density. Moving on to the non-COPD cohort, none of the multimorbidities that we looked at were statistically significant, but we definitely still see that signal trending towards the same. And I will say the only multimorbidity that was significant was the aortic calcifications, which I think is not all that surprising.
Yeah.
Yes.
Maybe I missed it on your previous slide, but these are matched by smoking, both of these groups.
So this is tobacco exposed cohort, correct? Yeah. The phase 1 data only had about 22 non-smokers, and so that's and I agree, that there is probably some bias associated with that, but that's kind of the cohort that we're working with right now. Yeah. And then our other outcome that we were interested in is mortality. So this is a forest plot. I'm going to take you through the axes and then the data. So you can see that we have the models on the left, and then we have the hazard ratios, and then also the significance of those hazard ratios. We have the entire cohort, COPD only, and the non-COPD, and I pulled out lung function as well. I thought this was really interesting, and I can't necessarily explain it to you just yet, but we found that accelerated aging does independently predict mortality for both groups, and you can see that based on the hazard ratios here, that it's actually independent of lung function. It's interesting, right? Because this is even in the COPD, non-COPD cohort, we see this. Interest and FEV1 remains a strong predictor of mortality in those with COPD. Interestingly, we found a stronger mortality signal in the non-COPD cohort. And the SOMA scan proteomics is plasma samples, so maybe we're finding more of a systemic whole body signal, or there are multiple competing risks that we haven't been able to elucidate just yet. But regardless, we see this. Regardless, we see the signal here in both the COPD and non-COPD cohort that's independent of lung function and is significant. So just to kind of summarize quickly the work so far, we've derived this 87 protein biologic age endotype and developed an accelerated aging score. We've shown that those with COPD have an accelerated age of about 25 years, meaning difference beyond their chronologic age. Pathway analysis shows cell signaling, ECM remodeling, and immune and inflammatory responses, some of the dominant biologic processes. And this accelerated aging metric does independently associate with multi-organ dysfunction, functional impairment, and mortality in those with COPD, and has a mortality signal even in those without COPD. Next steps, I'm working with some folks in spiromics, which is another multi-center perspective cohort for COPD participants with COPD. I think it would be really interesting to look at a non-COPD healthy cohort for validation. So talking about potentially, a MESA or some other cohorts. And then, I think it would be really interesting to look at some longitudinal follow-ups to examine adjudicated deaths, which I'm doing now in the COPD gene phase 2 and 3. And then also doing some lane factor analyses, looking to identify some subcomponents of the proteomic signatures tied to specific pathways, and then continuing that analysis that I briefly described earlier. Any questions on that before I pivot a little bit?
Any tiny questions?
Yes.
So I've always been bothered by the VOD index. For those people who don't know what it is, the B in VOD index is body mass index. And
Sure.
I understand cachexia,
Sure.
And people fading away, low muscle mass, increased mortality. But these are all developed on cohorts of survivors. Isn't it survivorship bias? If you are morbidly obese with COPD, you're dead and you're not in the cohort.
Yes. I mean, yeah, I see your point. I think for, the blue bloater, pink puffer, we actually see cachexia as a big prognosticator of mortality, and especially in those with emphysema. So, I mean, yes, I think either way on one side or the other, you do see that increased signal. I will say we adjusted for BMI. And so, Bodex index is not a great, it's not very specific, but, we included it because we had the data, but we did adjust in terms of for BMI and some of these other confounders.
Well, it's been out there, 20, I think it's 2004, something like that. I mean, it's kind of standard, but I've always wondered about it.
Sure.
And we just have such a large obese population of smokers here. It just worries me we're missing how risky that is.
Yeah, absolutely. I will say in some other work I haven't shown here that I'll briefly discuss in this project, we do see central adiposity specifically and associated with this pre-diabetic endotype in more of a emphysema than airway actual. So we see this, and that's not new, right? Airway versus emphysema phenotype, but we see more of this metabolic syndrome with that as well, which is interesting. So I'm going to shift gears a little bit and just discuss a separate but parallel related project, looking at microbial translocation and alveolar dysfunction in COPD. So dysbiosis can disrupt, dysbiosis is an imbalance in the microbiome. It can increase epithelial permeability in the gut, driving microbial translocation or the movement of bacterial and fungal products across an impaired intestinal epithelial barrier. It can, it has a wide systemic effect, but also causes chronic low-grade inflammation, which we know is a driver of aging, as I've re-shown you this figure down here. In COPD, we know that circulating fungal products in the absence of active infection associates with decreased lung function and clinical outcomes marked by some frailty measures. And data on microbial translocation and multimorbidity in COPD is scarce. So just some validated measurable in vitro, surrogates of microbial translocation include beta-2-glucan, fungal cell wall polysaccharide, LPS, bacterial endotoxin, as well as intestinal fatty acid binding protein, IFABP, which is released by damaged enterocytes and used as a surrogate of impaired epithelial barrier function. So we were interested in microbial translocation contributing to multimorbidity in COPD. So what we did was first, we looked at intestinal fatty acid binding protein within an internal cohort at Pittsburgh called the SCORE cohort. We measured plasma levels of intestinal fatty acid binding protein with ELISA, And what we found was that expression was increased in those with COPD compared to those without, and significantly associated with multimorbidity, characterized by emphysema and decreased lung function, this metabolic syndrome characterized by central adiposity and prediabetes. Vascular disease characterized by aortic and coronary artery calcification, and this musculoskeletal dysfunction characterized by decreased bone density and increased intramuscular adipose tissue volume. So just to kind of recap really quickly, dysbiosis, microbial translocation related to aging through chronic inflammation, microbial translocation of beta-D-glucan and intestinal fatty acid binding protein associated with decreased lung function and multimorbidity and COPD. So we're wondering if there was a mechanistic receptor that could contribute to this process in pathogenesis. So I'm going to delve into a little bit about receptor ephrin A2. It's been mostly studied more in the oncologic field. So there actually are already FDA-approved drugs targeting this receptor, which is receptor ephrin A2 is an epithelial surface receptor. It functions as a pattern recognition receptor for both beta-D-glucan and LPS, both in the gut and in the lung. It activates NF-kappa B and has a strong pro-inflammatory response. And so we looked at some single-cell RNA-seq data of human EpCAM epithelial cells from COPD and healthy donors. And then, as you can see here, this is the single-cell RNA-seq data. Ephrin A2 is enriched and upregulated in COPD AT2 cells here, compared to healthy AT2 cells. And then ephrin A2 was further preferentially expressed by basal cells, actually, as well, here, compared to healthy. And so, to confirm that ephrin A2 was blockable when treated with beta-D-glucan, and that it was a beta-D-glucan receptor, we exposed human bronchial epithelial cells, BSTBs, in vitro in monolayer to zymosin A, fungal beta-D-glucan, at 100 mg per ml for 24 hours with increasing concentrations of ALW, which is an ephrin A2 blocker. Then we collected the supernatant and looked at cytokine and chemokine expression. So what you can see here, none for both. When we treated the BSTB cells with beta-D-glucan, you can see an increase in IL-6, IL-8, CXCL5, and TNF-alpha. And then, interestingly, and as expected, when we treated the cells with this ALW in increasing doses, as you can see here by the pluses, that we were able to block the cytokine-chemokine response in a dose-dependent fashion. So next, we want to confirm with immunofluorescence staining to look at the expression and localization of ephrin A2 expressing cells in human tissue with AT2 cells. This is just a representative staining here that I've thrown up. The top is healthy donor, the bottom is COPD tissue. So we're going from just ephrin A2 staining to ephrin co-localized with AT2 in the yellow, and then a merged photo with the DAPT in blue. What we're finding is that, we do see this signal. It's not statistically significant, but I'm still going through the samples. So there's more work to be done here for sure. Going to do some formal quantification of ephrin A2 expression and localization in AT2 and basal epithelial cells. And the plan is to integrate some bulk RNA-seq data as well to define ephrin A2 related pathways in COPD. In summary, circulating microbial and fungal products associated with decreased lung function and multimorbidity, potentially accelerating biologic aging in COPD. Ephrin-A2 is upregulated in COPD epithelium and may link microbial translocation to epithelial inflammation and remodeling. In vitro, we saw that Ephrin-A2 blockade does reduce the cytokine and chemokine response to beta-D-glucan, potentially identifying a therapeutic target. So I want to take a step back a little bit and talk about just kind of big picture, overall future directions. I'm really interested in integrating proteomic and mechanistic studies, linking microbial signaling and biologic aging trajectories. Of course, we have to validate the proteomic endotype, and interesting to see that longitudinally, and then to potentially identify biomarkers of multimorbidity and mortality in COPD. And of course, the overall goal is to advance clinical translational research towards more precision-based interventions. So thank you so much for having me. Couldn't do the work that I do with my mentors and collaborators and supporters. And these are just some really important people that I've worked with.
I have one question.
Yes.
I know it must not be very relevant. You just said it about beta-D-glucan and LPS. So I just wanted to know what kind of patient population I think COPD versus non-COPD. So if I go very sick, hospitalized patients, or just outpatient cases, like what kind of patient population? Very sick population.
So I'm just
selected this.
So I just want to repeat your question to make sure that I understand. You're asking for the first part, the people in which beta-D-glucan was measured and found to be elevated in the absence of active disease. Who were those participants? Okay, it was in an ambulatory setting, actually within our own cohort at Pitt.
Okay, yeah, thank you.
Mm-hmm.
Can I ask a question? And if this question sounds simple or stupid, I'm just
There's no such thing. No such thing.
When we talk about lung aging as a process, is it primarily driven through inflammatory pathways or degenerative pathways like apoptosis, or is it an impaired repair, or all of the above?
Yeah, so I think that's a question I would love to be able to give you a very specific answer to right now. I think you're kind of at the crux of it, right? And I think part of the issue is that COPD isn't necessarily one disease state, one phenotype, right? And so that's why we're trying to get to the endotyping. But I think what you can see from our pathway analysis that we did, there's a strong ECM signal, which I think is not unsurprising given some of the histological changes we see with emphysema. So it seems like there's a strong inflammatory signal, but is that downstream because of ACM remodeling and some of the other cell signaling? Not entirely sure yet.
The loss of elastic fiber that happens as a normal part of lung aging, right? Is that physiologically different than the loss of elastic fiber that you see in emphysema-related aging?
I think,
or is it just the extent and acceleration of it that makes it different?
So it's I'm not an expert, but my understanding is that sometimes we can use senile lung tissue actually, to also look at architectural and tissue changes. So I think, and if anyone knows the answer, please correct me. But my understanding is that it's likely more of an advanced process.
But at the core of it, it's just the same physiologic process, but just it's revved up in a way.
Potentially, but we also don't know why some patients with emphysema, advance with very minimal exposure versus others who, maybe smoke their entire lives and never. And so I think there's potentially some, genetic susceptibilities.
I wonder, we see a very heterogeneous COPD population. We will have, I remember patients with FEV1 in their teens who are still riding their bikes and going to bike rallies. And you have patients with,
absolutely.
And yes, there may be many factors that create that phenotype, but I wonder if the final common pathway is different types of aging.
I think so.
That lead to
exactly
different
so I think, this is not probably applicable to all patients with COPD, but I think the goal is to potentially identify a subset of accelerated agers who are more at high risk for developing some of these multimorbidities potentially at an earlier age, because those are the patients that are going to have, you know, the worst outcomes. Yes. Dr.
Another
Williams.
question. I always have the nutrition question. This one is, you know, giving these lectures on cardiovascular disease and started looking at, you know, planetary health
Sure.
diets. This is a big deal now. They've got a journal about it and everything. And among the reduction of hypertension, diabetes, obesity, inflammation, and hyperlipidemia pops up, and inflammation suddenly pops up respiratory death, lung disease mortality, mostly from COPD. When you look at the data, there are a couple of cohorts, one in the US, one in China. Well, actually, it's Singapore. Publication came from China, but they are talking about a 20% reduction in respiratory mortality if people follow the Planetary Healthy Diet Index, which is basically getting rid of animal products. The problem I have with it is it's all observational. Sure, it was 23 years, it's outcome. But that to me is just hypothesis generating. So I've looked, I haven't seen anyone take a set of COPD people and then do a dietary intervention. And we know it's going to lower inflammation, and that mechanistically could, and it also changes the microbiome, which you've elucidated really well. So, you know, what I'm wondering is why the field, since I'm not a pulmonologist, why is anybody going after this? Or maybe
You need tissue. You need tissue. Yeah, I mean,
well, could you follow lung function decline over time, a long cohort like they did? I mean, that was 20. We're not going to do 23. Well, we could do 23 years because you're just starting. Okay.
there's some interesting data looking at, there's a study, there's a cohort in Baltimore at Hopkins where they looked at saturated fats in people with specifically a BMI of over 30 and lung function, and I think they actually found some protective effects. So I think some people are interested, but it's not my area of expertise. So it's certainly interesting as a contributing factor.
Sounds like an RO1.
You know, Dr. Williams, there's a plethora of plant-based supplements out there. You know, non-hallucinogenic mushrooms, and, you know,
Oh God, yeah.
I mean,
Since I was in
you
clinic.
can put your finger and they're out there. And they all claim that they'll reduce your aging and do all these wonderful things. And I wonder if anybody has ever looked at it, even in vitro.
Right.
Hallucinogenic mushrooms.
So you may not be any better, but you won't mind it one way. So the real question is, has anyone actually done this as an intervention?
It's an intervention
for COPD, and look at the parameters for COPD.
Yeah, or lung aging in general. You know, can slow down.
Lung aging, I think it's becoming a little bit more popular or more well studied, which is exciting. But most of the aging, you know, even if it's organ specific, has been based on plasma and whole blood, which I think is a limiting factor because we know that organs age differently, different organ aging contributes differently to systemic aging. And then, you know, one of the things I'm interested in also is we know that the lung has multiple compartments. There's epithelial, interstitial, endothelial. And so I think depending on the exposure and where the exposure is coming from, potentially those could have different cellular effects as well.
What is the biggest obstacle you found in studying lung aging? Is it the availability of tissue, or is it the separation of these different processes? Because lung is a very immunogenic organ.
Both. It's there's a paucity of lung issue, and you know, eventually it'd be great to be able to kind of elucidate a more applicable endotype, right? Kind of incorporating potentially labs that patients just get on a daily basis. And some people over at the UK Biobank have been trying to do something like this. And then, you know, somehow integrate that with, I think, EHR data as well would be really interesting. But if you want to look at lung specific, I think, we need tissue, as well. So,
We know now that patients that have COPD are colonized with bacteria. They tend to have frequent exacerbations. In fact, I'm now checking sputum on those patients that are compliant, that have a good diet, keep getting exacerbations, especially if they're colonized with Pseudomonas and H. flu to eradicate them. On those that you look for the translocations, were they colonized?
we did not have that, but that is something that, one of the mentors is looking at. Yeah. Getting like nasal swabs and other things, yeah.
So could I follow that? So you talked about the microbial transfer and you talked about dysbiosis, but you kind of had multiple figures linking the gut and the lungs. And so when you do things like looking for LPS or beta D glucan, how are you separating out where the site of that translocation happens? And when you think about dysbiosis in the lungs, are you simply thinking about pathologic organisms that happen as a result of decreased clearance, or is there some other marker? Because
Are you
dysbiosis in the gut is relatively well defined. I don't know that in the lungs is.
It's not well defined in the law. And so, I think that is one of the weaknesses, or one of the things that we're trying to figure out. I think it's a chicken and egg question, but we want to first establish that this process is happening and then delve a little bit more into the detail there. But yes, agreed. Chicken and egg question there.
What about the translocation, lung versus gut? Like, how do you differentiate that, or does it matter?
I think it does matter. Dr. Bond, one of my mentors, is kind of in the process of trying to, collect samples from various parts of the tract, and then we'll be looking at this. And this is some more preliminary data for part of that work that I've been doing.
Are we assuming that the heavy smoker will not have COPD? They will have a normal aging then, and I have the accelerated aging.
I think that maybe, maybe. So I think, this endotype is potentially applicable to those without COPD if they present with some of these multimorbidities. But if those patients are, don't have any of these other things that we looked at, I'm not going to, I can't necessarily say that the score is as applicable or that they're going to be aging more quickly or not.
Needing tissue, I think there's hope, because photon counting CT is out there and it's coming here, and there are some studies characterizing COPD and chronic lung disease in very much detail. It'd be great to correlate it with the aging biochemistry and the microbiome that you're doing. We're apparently going to get one, and hopefully it'll be the next year or so. For people who haven't seen photon counting CT, just Google it and you will see it. Basically tissue histology done non-invasively. It's pretty amazing. Thank you.
I had a question about your proteomics. You probably said, but I missed it. I assume that was plasma proteomics.
Yes, right.
And so it's not surprising you saw ECM and inflammatory signaling. I was wondering if any of the proteins you identified were bacterial proteins.
So no, not that I know. No. Yeah. It is plasma, but it's from a COPD cohort. So we thought that we would really find potentially more lung specific signals. And I think potentially that's one of the reasons why we found this correlation with mortality in a non-obstructed cohort as well. So I think, it could be potentially interesting to do. We have to parse down the 87 proteins first into something a little bit more feasible, and then potentially do some more mechanistic work with that too.
Thank you.
Interested in that group of people whose biologic age is younger than their chronologic age. What is their secret?
I don't know, but I would be interested.
Maybe you should study them and find the fountain of youth. Huh? All right. Thank you so much.
Thank you guys so much.